this is a very quick cleanup lecture. It's a mini lecture. I want to remind you that instead of watching this video, you really should be watching the one on YouTube that I posted in the class Zoom chat. If you need that link somewhere, well, if you just type my name in YouTube, you should find my channel. And then I have a playlist for Bio 121, and then you'll see DNA replication and repair there, or you see like DNA for majors. So watch that. Okay, so the things that are going to cover in that video, we're going to talk about DNA repair. Okay, and again, for the test, I want you to know these two DNA polymerases for prokaryotes. Those are the most important. All right, and just understand the others are involved in repair. You don't have to worry about the details. And then for humans, I told you don't have to know all these, right? Know alpha. Everybody should know DNA polymerase alpha. That's the main DNA polymerase in eukaryotes. All right. And then just keep in mind that just like in prokaryotes, right, there's really delta is for the lagging strand, and then all the others deal with various kinds of damage. Okay. The most common kind of DNA damage repair is called nucleotide excision repair, where we pop out bad ones and put new ones in. And that's what we went over in the quiz earlier, the nuclease, the polymerase, the ligase. Understand that damage to DNA is not a mutation. So make sure you get this, right? So a mutation isn't damage. The damage happens, the damaged piece of DNA, that area, like right here, we would call that a lesion. Okay, and if the lesion is not fixed correctly, then it becomes a mutation, right? The key to mutation, it is heritable to the next generation of cell. Okay, and we'll talk more about mutations later after we do gene regulation. All right, so there's different kinds of repair mechanisms. You don't have to know the details of these, but really one important thing is we have, and this number is probably even bigger since the last time I updated this slide, but there's over 170 different enzymes involved just in DNA repair alone. So that should give you the notion that this is a super duper important thing. Okay. All right, I said all this already. But the question is like, what role does mutation play? We know that mutation is the key to evolution, right? Without mutation, there is no new stuff. Right, we learned about independent assortment. We learned about crossing over, right? But without anything else, right? If we just have the same cards to play with every time, we're eventually going to run out of choices as the environment changes, right? So what mutation does is it gives us brand new cards, right? And there's all sorts of different kinds of mutations. And we already talked about, chromosome level mutations. Think of translocations and inversions and deletions and all that. And then there's something called a point mutation. And we'll learn about more of those when we do the genetic code. Okay. Somatic, you should know that word. Somatic cells are body cells, right? Somatic mutations, therefore, would be mutations that affect body cells. Somatic mutations cause cancer. Those are the cancer-causing mutations. Those are the ones that you get in your somatic tissue, and you don't pass those on to your kids. Germline mutations are the ones that get passed on. Germline mutations happen during gametogenesis. Remember oogenesis and spermatogenesis, right? So mutations during that process, those are the ones that are important for evolution. Okay. Now telomeres, again, very briefly, telomeres are the ends of chromosomes, and they're really hard to duplicate. Right, the idea conceptually is this: if this is the literal end of the chromosome here, let's say this is the end of the chromosome. Remember that as that replication fork proceeds, right, that leading strand isn't going to have a problem, right? The leading strand can just go into the end. The lagging strand, though, let's just separate that bottom strand out here, right? We have to go here and then build this way. Then we have to go here, and we have to go this way. We have to go here, we have to go this way. And oops, so let's say there's a little extra here. I can't add a primer here, right? So there's always going to be this little extra that I can't replicate. So you don't. So what happens is, over time, the end of a chromosome literally gets shorter and shorter and shorter until it starts cutting into a key gene region, whatever is there. And so to get around that, what cells do is they have this sensing network. They have the ability to sense this. And once a telomere gets to a certain critical length, whatever that length is, the cell will stop growing no matter what. It will refuse to listen to commands. It will refuse to respond to growth factors. Those are called senescent cells. A senescent cell is a cell that will not go. Okay. So there's different kinds of senescence we're not going to get into. I'll just tell you that cells that are senescent are cells that are no longer able to grow. And it's a protective mechanism, right? You don't want to grow if your telomeres are that short. Okay, that's all I'll say about that. Okay. I don't know, you guys are, some of you are a little too young now to remember Dolly, but Dolly was the very first cloned animal. Dolly was, so what you basically do is you take an egg cell. Okay, so here's a human, or here's an animal oocyte, right? You enucleate it. You literally get rid of the nucleus. You can go in there with a needle and suck it out. Okay, and so what do you get? You get a cell without a nucleus. Okay, but it's an egg cell, so it has all the other good stuff in there. And then what you do is over here, you get some kind of random, let's say a skin cell, right? This nucleus, that's a diploid nucleus, right? That's a 2N nucleus. We enucleate this cell and we transfer this nucleus into here. So now this is a 2N cell. And then we stimulate this thing with growth factors somehow. And that coaxes this, which it kind of looks like a zygote now. And it starts going through the stages of development. That's how we clone things. So Dolly was born in this way. The problem is that when Dolly was born, she had the telomeres of her mother or herself. She's a clone, but she had shortened telomeres. She started developing age-related diseases after being a year old. And she died at about, I think, 4 of old age. And if you click on that link, you can read a story about her there. Okay. This is how telomerase works. You can watch the video on that. But the idea here is it extends the telomere artificially. Or not artificially, but it extends it when it otherwise wouldn't. Okay. Here's a slide on senescence. There's the idea of permanent. So senescence is defined as permanent cell cycle arrest. That's the formal way of saying it. And this is a very alarming stat. Okay, almost all cancers have elevated levels of telomerase. When we first discovered this gene, by the way, we used to think, so this is back in the late 90s, early 2000s. So I was in grad school when it was discovered. I remember one of the professors kind of getting excited about it. Like, this is the key to immortality. They were able to make senescent cells. They were able to take senescent cells, turn on telomerase and make them immortal. So we were taking normal, non-mutated, non-cancer cells and pushing them into the cell cycle, which was really exciting work. But of course, everything is too good to be true when it looks that way, right? And so it turned out that, it was shortly after that discovery, they realized that almost all cancers also have it mutated. So if we took a pill or we had a virus infect us or whatever, and we were able to turn our telomerase gene on, all that would do is accelerate the pace at which we get cancer. So no fountain of youth here. Okay, I want to spend some time on this because this is going to lead into gene expression. Okay, you guys still with me here? We're gonna, so this is important. Raise your hand if you're listening on. I'm not sure where you guys are right now. Okay, we're all here. Okay, so have I showed you my piece of paper yet? Have I brought this out? This is one of my most important props. It's just this was a piece of junk mail I got last semester, and I crumpled it up. And instead of throwing it away, I realized this is a perfect way of explaining how DNA works. Watch this. It's crumpled up. And now it's open, and you can read it. And now you can't. Okay, that's the way this works. So there are these proteins called histones. We talked about briefly when we did this couple of months ago, but histones allow DNA to form these higher orders of structure, right? Remember we talked about supercoiling with my other great prop here, right? We talked about the idea that DNA is under this incredible amount of supercoiling, right? And so the interactions here with histones control how dense DNA or how fluffy DNA gets. When we talked about heterochromatin and euchromatin. Okay. All right. So as a refresher, so go back to your notes from the last time we did this and look along here from so we have our naked double-stranded helix. Then we have our beads on a string, right? And so those individual circles here represent the nucleosome. And then we start twisting this, and we start to get what's called the solenoid, or the 30 nanometer fiber. And if we just keep going and supercoiling the supercoils, we get what are called chromatin territories, which are like continents. Okay, you can also go in chromosome territories. So if this is a nucleus, you'll have some region of the chromosome or some region of the nucleus where each individual chromosome occupies a defined space, like continents on the Earth, except in 3D. It penetrates down into the nucleus too. It's not just on the surface. And we call that overall structure the fractal globule. Okay, and wow, that's a typo. Chromosomes, that's chromosomes. Chromosomes. All right, and so mitotic chromosomes are this special use case, right? When we're going to move everything along and package them. So here are your nucleosomes. All right, know your histones. You got in the octamer itself, you have two H2Bs, you have two H2As, you have two H3s and two H4s. All right, so these are all times 2, and you get the octamer. All right, so let me write those in order: H2A, H2B, H3, H4. That forms the octamer, and then the linker in between them is H1. Right, so this was me drawing two nucleosomes next to each other. Right, so the blue here, there's your H2A and B, H3, H4, and then you can see that this linker histone H1 helps stick it all together. Right, and so you can see carefully here, here's the double strand of the DNA. So the DNA itself goes around each one of these twice, like this. Okay, and so you can see here on this picture here, you can see the double strand of DNA. All right. All right. Now, you see right here and here and here, do you see these little things sticking out? Those are called histone tails. Those are important here. And so I want you to keep this in mind as we do gene regulation later. Okay, these are positively charged. This is what allows the DNA to stick to it, because the DNA is negatively charged. Okay, and so one way to regulate all this, ready? One way to regulate all this is to take this positively charged histone tail and to cover it with a mask. Okay, and that is called acetylation. So I shouldn't say it like that. I should say it like this. In order to mask that, we acetylate it. We acetylate the tail. And what that does is it neutralizes the charge. Okay, and so there's a yin yang here. We can put the cap on, we can take the cap off. Okay, so there are proteins called histone, acetyl transferase, or a HAT. Right? How do you do? HATs are good things. Histone acetyl transferase. They put the histone into a state that is no longer able to interact with DNA. They open the DNA up. So you can think of these as positive regulators. They will be gene activating. And then the opposite is called a histone deacetylase. And we abbreviate that HDAC. Okay, so you can simply put it like this. You have hats that activate genes, and you have HDACs that would inhibit. And you could even say gene expression. Okay, so this is one of the most fundamental ways we can regulate genes at the highest levels. Hey, Jordan, I just, you're making me feel old, man. All right.
I knew you were gonna say that. I just found it really funny
It's
because he
really
was
funny.
walking up on the stairs in the middle of it. He was like, "Oh," he just started rambling. I was like, "What are you talking about?" He's like, "Dolly, I remember her." And I was like, "Oh, okay."
Yeah, it was like 1990, 1997, I think. Anyway, okay, so that's histones. This is what it looks like. This is actual X-ray crystallography data. Remember Rosalind Franklin? That's what the kind of stuff she worked on. This is a modern version of that. This is the kind of pictures we get out of it. Okay, so you can see here in the aqua or the teal color, that's the DNA wrapping twice, right? So you could see the DNA is here or the DNA is here. So this is kind of a 90-degree tilt. Okay, and you can see the linker coming out here. That's the H1. This is H1 over here. And you could see, if you're really careful, you could see the tails. Right, so that's probably a tail. That's a tail. That's probably the tail. Right, so the little things that jut out of it. All right. So there's a nucleosome, beads on a string, and we're still at a level of understanding this. We're still studying this stuff. So this used the speculative model from 2002. The idea of like, how is this arranged in 3D space? We figured it out since then. And it turns out that archaeans also have histones, but they organize their DNA in a slightly different way. Whereas bacteria do not have histones, they have other proteins that are homologous to histones, but they lack the H1, the H2s, and the H3s and the H4s, whereas archaeans have them. Okay. And then this is an important picture. I want you to look carefully at this. You can see the beginning of what's called a looped domain. So you can see that we can put some genes out into space where the genes can be active. They can be active, right? And then we come back in here, and the rest of this is just tight DNA, right? So we can basically picture like a ball of yarn and then pulling a thread out of it and then being able to read through that. Okay, so that's the idea here. And then I took this picture in grad school. This is one of the things I was very proud of. This is the nuclear matrix, supposedly. And the more I stare at this, the more I realized that I was probably looking at chromatin territories. So this is a protein called PML that I'm doing immunofluorescence with. Actually, Julia, your boyfriend probably want to look at this. This is one of the things that I was working on.
He's not here right now, but as soon as he's here, I'll show him the PowerPoint slide.
Yeah, okay. So yeah, this is staining, immunofluorescence staining with the anti-PML isoform 2. I overexpressed it with a plasmid, and then I looked. And I saw this. This blew my mind. I still remember it was a Friday night at like 2 in the morning when I finally saw this for the first time. So the idea is the nucleus itself may or may not have a proteinaceous filament network, like the cytoplasm. All right, it's been long thought it doesn't have it. And it still might not right now. I kind of checked out a couple years back, so maybe somebody's done more recent research in this. But the idea is that so it's called the nuclear matrix. It's usually one of the ways I see it is because the nucleus is so dense and under so much pressure, basically, that in between the chromosomes, I want you to think of that space as canals. Okay, so I want you to start seeing the nucleus in your head. If you close your eyes and imagine this, you have these three-dimensional blobs of chromosomes. In between them, okay, so if I was to doodle it, in between them, the space here, think of those like canals, like Venice. And those canals are filled with protein and RNA moving. Okay, there's two types of traffic. There's traffic coming and going. Okay, so think of the nucleus as a busy metropolis, and there's this constant movement of things coming in and out of it 24/7. It's like New York, the city that never sleeps. Okay, so beyond this, you don't have to worry about it. This is all hypothetical stuff now, but this is one of the areas that's most exciting to me in research, because understanding the nucleus helps us understand almost everything. So there are these things that we sometimes call TADs, topologically associated domain, that are basically huge regions of genomic DNA that act kind of like islands with each other. So the idea here is we have a higher order of organization well beyond the nucleosome, well beyond even the chromatin territory. Okay, and we're still not really understanding this stuff, but we do notice that in some diseases, we notice that this kind of interaction is disrupted. Okay, so this level and beyond is still a topic of debate in the current, like this is current level research that people are. So if you like this stuff, this would be going to molecular biology and then going to nuclear architecture. And there's several groups of the country that study nuclear architecture. One's in Cold Spring National Harbor, Cold Spring Harbor National Lab up in Long Island. Okay. And I'm gonna skip all the rest of this stuff, but there's your fractal globule. There, each chromosome gets its own color, and you can see the pretty picture there. That's pretty amazing stuff. Okay. And if you're not sure what a fractal is a mathematical thing. Fractals are they're scale-free geometry. If you go to Google Earth and you look at the coastline on the East Coast, and it looks, like this, and then you zoom in tenfold, it still looks like that. You zoom in tenfold, and it still looks like that. And you keep doing that. That's what a fractal is. That's the concept of a fractal. The most famous one is called the Mandelbrot fractal. That's what this picture is up here. So if you go to YouTube and you type, let me see if I can spell this right. I probably can't. It's a Mandelbrot. I think that's how you spell it. So you type Mandelbrot fractal, and there's some really amazing animations on it. Anyway, that's that. I taught you this earlier. I'm going to say it again. There's two types of chromatin within the nucleus. Okay, these are pictures I took too from my old research days. This is DAPI staining, DAPI. It's a blue fluorescent molecule that intercalates into the double-stranded DNA. So if it can't get access to the DNA, you don't see it. So you see this dark region here? That would be the, you tell me, what would that be what's in the nucleus? What's a big ball chromosome, chromatin in the nucleus? What organelle is that?
Nucleolus.
Good, the nucleolus. This is the birthplace of ribosomes. Okay, you notice there's a couple regions of it here. These are U373 cancer. These are cancer cells. These are glioblastoma cells. That's why they look so disturbed. Notice the nucleus has this really funky shape to it. You see the weird shapes of these nuclei? That's one of the dead giveaways of cancer cells. Healthy cells never look like that. Healthy cells always have a nice shape like that. Okay, like that. Even that's not even, that's it looks more like that. This is, sorry, I was at the end of the PowerPoint. All right. So the euchromatin hold on. This actually has a typo on it. Just give me 1 second. I want to look something up really quick. Yeah, that's drives me nuts. Okay. Where's the picture? Okay, euchromatin. No idea why these were reversed when I added this heterochromatin versus U. Right, if you printed this out, I need you to make this typo fix. Okay. All right, so when you look at this slide, the bright stain is bright because the DNA is open and can get access to the stain. That's why it's bright. So the bright areas are euchromatin down here. Okay. And, all I really needed to do is change bright and dim, not the words themselves. There we go. Sorry, last update to that. Okay. The euchromatin there, the bright areas, not the dim areas, it's less compact, it's more open. All right, so what you associate euchromatin with, open, positive, you know, it's the good areas. It's the areas that are the active areas. Okay. In contrast, the dim areas, the dark areas, are the areas of the heterochromatin. That's where the DNA is tightly coiled up to the point where the stain can't get to it. Okay. And you can see just from looking at these pictures, if you just look at these pictures here, you can see that the nucleus itself is a very complicated place, right? The DNA has a very higher order complex structure, but at the end of it all, the simplest way you can chunk it all out is by saying there's dark and light areas. Okay, there's closed areas, there's open areas. Now, just because it's open doesn't mean the genes are active. It means that the genes can be active. So this would be like taking your toolbox from your garage, pulling it out, putting it up on your tool bench, opening it up, and just leaving it there. We can use it when we need it. Right? So you can pull the hammer out. We do. Whereas if it's packed up and stored away, you're not using it. Okay, so just because the chromosome's open, it doesn't necessarily mean all the genes on that chromosome are turned on. That makes sense? No? Yes? Am I losing you guys?
No, I understand.
Okay. All right. And so this, you study this slide right here. This would be the good one as a good summary for gene or for chromosome organization. Okay, and so I'm going to stop this recording. I'm going to pack this away. This is the end of DNA structure and function. And we're going to do gene regulation now.
Wait, excuse me?
Yeah, go ahead.
As part of the DNA packaging section on your study guide, you said you talked about epigenetics and genomic imprinting as well as DNA methylation. What should we know about those guys?
So I'll talk about that in gene expression and gene regulation. Yeah. Okay. All right. So I'm going to stop this recording so you guys have a neat tie off here.